As we enter the fourth year of this series, looking back, we have come a long way. We have put together some very, very good events. And it has fulfilled the objective that the Advisory Council set out for these events, that was to inform the community, both within the university and outside, of topics that have societal impacts. Around the same time that we started this, the National Academy of Engineering issued their list of grand challenges for engineering. A dozen topics appeared on that list. And it was interesting that everything came together. We have been arranging these events around those grand challenges. So welcome to this event, and very pleased to have you with us. Let me acknowledge help from a number of folks who have worked hard to put this together. The organizing committee, chaired by Nick Ceramic, who is also the moderator, with committee members Susan Fort O'Neill, Ken Peterson, I don't see him here, Mike Nigley, and Matt Petrim. Thank you all very much. I'd like to acknowledge Mike, who, as I said, Mike Nigley, who was the chair of Advisory Council at the time and also chaired the organizing committee for several years, and now is retiring as the COO of the San Diego Gas and Electric. Michael, we wish you well. And thank you, organizing committee, for your hard work to put this together. Other members of the Engineering Advisory Council who are with us tonight are Barbara Barkan, Chris Hernandez, I just saw him walk, yes, sitting in that corner. Dan Craig, where are you sitting? Okay, great, thank you. And thank you, Advisory Council members. The sponsors who have made this possible are Applied Medical, San Diego Gas and Electric, P2S Engineering, Port of Long Beach, Fort Design, and Technolink Association. Thank you for your sponsorship that makes this event to happen with your generous assistance. I'd like to acknowledge presence of several of my colleagues, including the Dean of the College of Business, Michael Solt, standing in the back, and our Associate Vice President for Research, Andrew Mason, sitting in the audience somewhere. Thank you. I'm sorry if I missed someone, but this is a dynamic list that I've been implementing as evening goes on. So on to the topic of tonight's lecture, Engineering for the Body. Interesting topic, and we can all relate to it. How many of you have a dental implant? Oh, only a few. Oh, you're shy. Come on. Well, that's the probably the most ancient form of engineering for the body. It goes back to the time of ancient Egypt. And we have come a long way. You know, who has a hip replacement or an ankle replacement or whatever? Well, all of those are things that have come about in the recent years. But really, in the 21st century, we have seen things move forward quite substantially. For example, with 3D printing. That has opened so many doors for us. Bioprinting, well, you know that, yeah, we can do prosthetics and so on. Well, the ink, what's the ink for that printer? It's some form of composite. Well, the same technology can be used to do DNA printing. What's the ink for that one? Cell. Well, that's layman's description of this very, very deep and important technology. But really, we have synthetic biology has brought the tissue engineering to really the digital age. And we're going to hear from the panelists on this topic in different perspectives that they have. And the moderator for tonight's lecture is Nick Ceramic. I have a distinct pleasure of introducing him. He is a senior project leader in systems engineering for the Aerospace Corporation, where he has worked for over 30 years, primarily on military satellite systems. He is an American Institute of Aeronautics and Astronautics Associate Fellow and a Computer Systems Technical Committee member. In July 2007, Mayor Bob Foster appointed Nick to the Long Beach Board of Harbor Commissioners, and he represents the port on the Intermodal Container Transfer Facility Joint Powers Authority's Governing Board. I did it again. Well, this is probably the longest committee title I know I can recall. I'm proud to say Nick is our own distinguished alum, College of Engineering alumnus, earning both his bachelor's and master's degree in electrical engineering at the Beach. And he's also a member of the Dean's Advisory Council and chairs this organizing committee. Nick?
I guess this mic's on. Okay, thank you, Dean. First of all, I'd like to thank our great panel members. This is, we've been able to really get some of the top people in the industry to really come out here and talk. So I'd really like to thank you for taking the time with us. Second, I'd like to really thank everybody out here, because without you coming here, this really wouldn't be much of a lecture series. But the first thing I really wanted to do was let you know more about what we're doing with the lecture series. Just give you a little hint and then get into our speakers here. The Dean's Advisory Group and our lecture committee decided what we wanted to do was make both the fall and the spring topics under basically one umbrella topic. This time it's been, in fact, the last lecture, if you remember, was robotics, in robotics in medicine. Well, this one here is reproducing body functions. And this time we're going to be talking about body functions and everything. The next time, and I don't know exactly what the title is going to be, but what we're going to be talking about is modeling of the brain. I guess there's been a lot of research, a lot of work being done on that. Just going to be another fascinating topic. And so then after that, the next fall and spring, we're going to come up with another set of topics. This way we actually get to actually bring out more underneath that topic if we do 2 lectures on it. So it's really, I think, it's going to be good. The topic today, is going to be reproducing body parts. It's an excellent topic because of all the work being done, the research being done. I think it's kind of fascinating. Dean touched on it. I think some of this advanced research and advanced technology that's being worked on today, we're going to be hearing about. One of the things is tissue and tissue research and tissue reproduction. And think of the people who need this. And from a social standpoint, if nothing else, that it actually will correct problems that they have and everything. And so by moving all this technology, advancing the technology, just moving along like as fast as, it's unbelievable how fast it's going, because, this topic 2 years from now may be old hat almost, probably not for you guys, but we're going to move into more and more new topics and new research that's being done. So this is just fascinating to me to get into stuff like the tissue engineering and some of those topics. Well, enough of that, Roy. Let's get into hearing more about the future of medicine, medical care out there. What I'd like to do is kind of challenge the students and the faculty out here, and even other people who really aren't students and faculty, is I always like to put this challenge out there, because I wish I had done this when during the time I went here, was to see something like this, and there are areas that you might want to do, move over into, do research in, do some work in. So think about what's going on here, and if there are areas you want to get into, maybe there are at Cal State Long Beach, they're really expanding programs and everything. So think about things that you might want to eventually get into here while you're still students. When you get like me, it's really You know, I'd love to go and change topics, change jobs, but it's a little bit late for me. So save some time. I've done this in the past. I've said that I will not read the speakers' bios. You have a book here. Please read those. It's just very, very impressive bios of everybody. So please go through it. Okay, and so I'd like to really get started right now. I'd like to introduce Dr. Adam Young, bioengineering consultant for cytograft tissue engineering, and he's founder and president of Wolf Biosciences. Adam? I think this is live too.
Okay. Great, thanks Nick for that introduction. I think this is on, yeah. So what I want to talk to you about today is adipose tissue engineering, and here we go. So we mentioned, so I have the distinct privilege of introducing this topic to you, and my background is in bioengineering. So bioengineering can range from prosthetics to medical imaging to even working with stem cells and some of those type of technologies. And my specific concentration is in tissue engineering. And what that is it's the application of both engineering and life sciences principles in order to help restore or improve the function of a tissue or organ. And in my case, I focus on adipose or fat tissue engineering. So you might be thinking to yourself, why would anybody want to improve or restore fat? That seems like a counterintuitive idea. Well, our focus at Wolf Bio is to help patients that are in need of repair of either a congenital or a traumatic type of adipose deficit. More specifically, we tend to focus on breast cancer patients. So as breast tumor detection gets better, these patients are now having a lumpectomy. So they have this tumor removed, and now they have a patient-specific void in that breast tissue that needs to be filled with something to repair the look. But this might also be the case for a third-degree burn patient. It could be somebody suffering from HIV-associated lipoatrophy, such as this gentleman here. Or even pediatric patients that might have some sort of congenital defect, like this patient here who's suffering from Perry-Romberg syndrome, in which she is unable to produce adipose tissue on just one half of her face, which you can imagine the type of impact that might have on her quality of life. As well, there are a variety of aesthetic applications, such as treatment of scars, treatment of wrinkles, those types of things. However, what's important about this is the products currently available for these patients are either synthetic or they are biochemically processed, such that within about 6 to 9 months, that material is completely broken down by the body, and the patient has to come back in. So imagine coming back to the clinic every 6 months, especially for a pediatric patient like this, where she has to do that for the rest of her life to try to maintain that type of appearance. So our question was, why can't we design a more natural material that will actually regenerate that tissue and give these patients a longer lasting effect? So within tissue engineering, one concept that's gained a lot of popularity over the past several years is something called decellularization. And it works almost exactly like it sounds. What we're doing here is removing the cellular content from an organ in order to isolate the extracellular matrix, or ECM, of that particular tissue. Now, the ECM typically provides structure for an organ, so I like to think of it a lot like the I-beams of a building that's being constructed. However, it also does a lot more than just provide structure. It also interacts with those cells. It provides them cues on a tissue-specific level of what those cells need to do and how they need to behave. So what we've seen in research over the past about decade or so is multiple organs have actually already been decellularized. So you can see a heart here. As it decellularizes, we're left with that white extracellular matrix-like material. However, none of these materials is very adipose or fat-specific. Again, additionally, all of these materials are sourced from animals, which can provide further biocompatibility issues if we were to put that into a human patient. So what our goal was we decided, hey, we've got all of these patients, especially in San Diego, that are getting liposuction, and that tissue is normally just thrown away. So why don't we try to use that for something else? So we figured, why don't we decellularize this human adipose tissue, and now we have a material that has adipose-specific cues, but it's also from a human origin. And fortunately, it's very abundant in San Diego. So how do we start with this? We have collaboration with multiple plastic surgeons in San Diego, We collect the leftover liposuction fat material from those guys. It's very gross. It's kind of like Fight Club if you've seen that movie, except everything's legal. We get patient consent first. So the material is processed for 2 days in sodium dodecyl sulfate. This is an anionic detergent, much like the laundry detergent you use at home. And what it does is it actually disrupts the cellular membrane. So it causes these cells to burst open and get washed away. We then process the tissue with lipase and colipase. These are 2 enzymes that the body naturally produces in order to break down your dietary fats. So we borrowed that same concept and processed our tissue here with those same enzymes in order to get rid of all of those lipids. So now we're left here with this white adipose extracellular matrix that's free of cells and free of lipids, which you can see in the staining here. An H&E stain would highlight nuclei as purple, so you can see that those are gone here. And the oil red O highlights lipids as bright red, and you can see we've efficiently removed those as well from our tissue. Now, I won't spend much time on the characterization. We've done a lot on it. But what I want to draw your attention to is here on the left side, we've done some immunolabeling. And what you can see is that our decellularized adipose tissue here in this right column has actually maintained a bunch of the extracellular matrix proteins that are specific to adipose tissue. So what you can see is not only have we removed the cells, but we've maintained those adipose-specific components that are naturally present in fat tissue. So our next question was, how do we get this adipose ECM into a patient? And we approached this by, we first dry out the material or lyophilize it. Then we chop it up into a fine powder, which you can see here. And then we wanted to get a liquid formulation of this adipose ECM. So we then digested it with the enzyme pepsin, and you can see we have this liquid formulation here. Now from here we can do one of 2 things. We can use this liquid to coat laboratory cell culture plasticware. So what this means is we can put this down on a surface, put some cells on top of it, and see how those cells interact with it. And I won't go into it today, but the publication down here, you can see how we've actually been able to show this adipose ECM can actually direct the differentiation of stem cells towards mature fat cells. But what I'm going to focus on today is what we found out was this liquid formulation of adipose ECM. If we bring it up to body temperature, so 37 degrees Celsius, it'll actually self-assemble into a soft hydrogel, much like when you put Jell-O into a fridge. So what this means is we can load this adipose extracellular matrix, this liquid formulation, into a syringe, and then inject it beneath the skin or subcutaneously, which you can see we've done here in a mouse. And within just 15 minutes, we get a nice soft bolus gel here that's sitting right underneath the skin. So this means not only have we isolated our adipose ECM, but we can deliver it into that patient and fill their specific adipose void. And it's not going to go, spread out everywhere throughout the patient. It's going to stay right in that one little spot. So our next step was to see, can this actually regenerate fat tissue? Because that's our goal. So what we did was we put this in a bunch of mice, and we came back at various time points, took it out, and looked at it under a microscope. That's what we do as scientists. So you can see here are cross sections of the tissue. It's been stained with H&E. And you can see here's the skin going across the top. Here's some subcutaneous fat that's there naturally. And all of this here is our adipose extracellular matrix that we injected beneath the skin. So at week 1, you can see there's cells migrating in. And week 2, same thing. But what's important here is we're not seeing any fibrous encapsulation. We're not seeing scarring. We're not seeing foreign body giant cells or other things that would be indicative of a negative immune response. So what this means is we have a biocompatible material. It's not creating some sort of negative immune response in the animal. But what's more exciting is at week 4, we actually saw a lot of pockets of natural adipose tissue that's developing within this animal. So if we zoom in here, all of these white voids here are adipocytes. It's very characteristic histology, which I'm sure is not the expertise of most of the people in here. You'd have to spend way too many hours looking under a microscope to pick this out. But believe me when I say that that's adipose tissue right there. So we not only are seeing this new adipose tissue developing within our adipose matrix, but we also stained this for a marker of blood vessels, smooth muscle actin, and saw a significant upregulation of neovascularization, or blood vessels moving into this material that we've implanted. So not only are we developing brand new adipose tissue in our material, but we actually have blood vessels coming in that are helping provide nutrients to those cells so they can continue to survive. So as an engineer, we established a set of design parameters that were important to us in terms of designing this material for adipose regeneration. And hopefully you've seen from this data that we were able to make a minimally invasive material, so we can inject it through a very small needle. We're actually, by sourcing this from human tissue, we can do this either autologously or allogeneic, meaning it goes from one human patient into another human patient, or from one human patient back into that same human patient. And then from our preliminary preclinical studies, we've also seen this is a biocompatible material, and it has actually caused the development or regeneration of brand new adipose tissue and even mature blood vessels that are going to help support that tissue as we move forward. So I think I have a couple more minutes left, so I want to change directions here just a little bit and tell you about some work I've been helping out with Cytograft Tissue Engineering as well. So Cytograft is a company up in the Bay Area, and their approach to tissue engineering and bioengineering is that they want to design materials that are natural, that don't use any sort of synthetic components or chemical processing. And they do this through a process called tissue engineering by self-assembly. So what they do is they take a small dermal biopsy, a little skin punch from their patient that's coming in. They then extract fibroblasts, which are a type of cell present in the skin that produce a lot of extracellular matrix, this ECM that we've been talking about, and then culture them in a laboratory and use them to produce sheets of human extracellular matrix. So now this is all lab-grown ECM, but they can grow it into a sheet. And what's great about this is then they can roll that sheet on top of itself to make a tube. And now they have a human-derived tube or blood vessel that they can then implant into patients. So you can see this vessel here. They can actually use that for bypass grafting. They can use it for hemodialysis access and a variety of other vascular applications. And what this does is it integrates very well with the patient's existing vasculature. So instead of having some other type of polymer stent or shunt in there, you now have what ends up being a healthy living vessel growing in there. So my work with them was to help develop another injectable form of this ECM, which they're calling BELV. So we used the same tissue engineering by self-assembly approach to have fibroblasts produce ECM for us in a particulate form. So these particles range from about 50 to 250 microns. But what's important here is the extracellular matrix that these particles are made of has many of the same components that were in our adipose ECM, these collagens, fibronectins, laminins. So we're seeing very similar compositional makeup of both of these materials. We then can suspend these particles in saline and deliver that through a syringe. So now we have another injectable material for treating these adipose deficits. So again, we went into animal trials. And what you can see here, so we have BELV on the top. Again, these are cross sections of the tissue. So we have the skin across the top, subcutaneous fat, and all of this pink material here are the injected particles that we placed in these mice. And we're comparing this to Juvederm, which is a cross-linked hyaluronic acid gel, which is, it's the most commonly used material for treating wrinkles in patients. So it's basically just a chemically derived gel that you can put beneath the skin. And this is 75% of the market almost. It's millions and millions of people get this put inside them every year. But what we're seeing with our BELV product, again, we get good integration, good biocompatibility. We're not stimulating some sort of intense immune response. But by 3 months, all of this white area here are new adipocytes, or new fat cells that are growing into our injection region. And this is in contrast to Juvederm, where we can see immediately by 2 weeks, that pink line right there is fibrous encapsulation or scarring. And it keeps getting thicker and thicker as we go out to later time points. And what this means is that the body has recognized this as a foreign material and is actually walling it off until it can totally break it down. And again, that's exactly what we're seeing here. So imagine, this is what's going on with all of these patients. They get this injected into them, and within about 6 to 9 months, the body's totally walled it off and broken it down. And what we're trying to do is design materials that interact with the body and actually facilitate regeneration. And if we zoom in, that's exactly what we're seeing here. So on our BELV product, as soon as 1 month, we're already getting new fat cells to grow into this area. So we're effectively regenerating this adipose tissue that's increasing in density as we continue out to later and later time points. Again, in contrast to Juvederm, which is seeing very negligible migration of cells in, we're not seeing any fat cell development, we're not seeing any of the blood vessels coming in. So again, those are the fat cells, and the black arrows are those new blood vessels that have migrated in. So just to sort of wrap everything up here, what we've seen is these naturally derived materials are really gaining a lot of popularity within tissue engineering. And in fact, our decellularized human adipose tissue provides a new novel material for these tissue engineering applications. And in fact, after we published this data on our injectable adipose ECM, several groups around the world have taken that work a little bit further to make different forms of this ECM. So we have beads, foams, various three-dimensional architecture, all made out of these biological ECM components. And finally, what we've seen is that we now have two different materials that have relatively similar compositional materials in terms of these ECM components, and they actually provide sort of a template for fat regeneration. So the cells that are migrating in recognize these components as adipose tissue or the scaffolding for adipose tissue, and think, okay, I need to now make some more adipocytes or more fat cells to repopulate this area. And what we're left here is potentially even a change in how clinicians will think about treating these patients. So instead of giving them a synthetic material that's just going to be broken down by the body within 6 months, we now have materials that can actually stimulate regeneration and provide that patient with a longer lasting treatment. So with that, I'll thank the many, many people that helped out with all of this research, as well as some of the funding sources. And I will be happy to take questions at the Q&A session, I think. So,
Thank you, Adam. Really interesting, I'll tell you, where we're moving today. Okay, next I'd like to introduce David Stout. David Stout's at Brown University right now, but he's a graduate of Cal State Long Beach here, and the beach. Okay, it's going to be a great presentation. You got some good videos, okay, hopefully.
Well, I hope so.
just keep going. I have a bunch of backups.
Okay. We'll get to that in the Q&A, I'm pretty sure. All right, excellent. Well, I have to start off by saying that this is somewhat of a surreal experience, because I remember the first time that the committee actually started when Dean Gulshani was, I believe, the second year as a dean. He actually started this distinguished lecture series, and I remember sitting actually in the seat thinking, man, those people are pretty smart. And now I was asked to come up, and I'm like, I don't know if I'm as smart as the rest of these guys, but I'm glad to be here. I am very grateful, and I'm really excited to present some of the new innovative research that's coming out of what we consider. Is this going? Oh. Can you guys hear me better now? Okay, all right. Some new innovative research that's coming out of what we consider the I-95 corridor, which is from Philadelphia all the way up to Boston. And this research is a collaboration between a lot of universities in that area, including MGH, Mass General Hospital, MIT, Harvard, Brown, of course, as well as University of Pennsylvania. But staying with body reproducing the functions, what I like to present on is a couple of ideas that have come out of this collaboration between these I-95 corridor research. And looking at kind of the major issues that are occurring in the healthcare world is you can think about is cardiovascular disease as well, and looking at a new innovative way to kind of think of from an engineering perspective, less than a tissue engineering perspective, but more on a biological engineering perspective of how can we apply material science, and mainly nanomaterials, materials that are on the 10 to the negative 9 meters, which has really been gaining a lot of attention in the last 10 years, and kind of coming up with an idea to kind of think of an alternative method or alternative way of helping individuals that have had a heart attack. And then next, looking at the other major things that are on people's minds, of course, is cancer. And coming up with a new assay or a new system from an engineering perspective so biologists and clinicians can see how tissue or how cells can migrate like a cancer cell. Not a lot of research has gone into understanding how the actual cancer cell migrates or metastasizes from one part of the body to the next. The American Cancer Association kind of has an idea how it goes through the vasculature system, but how it goes to the interstitial tissue, that's kind of still unknown. So if we can understand how these cells can migrate through the interstitial tissue or the tissue that goes in between the organs, then we can kind of begin to understand how the cell begins to migrate. So starting with cardiovascular disease or cardiovascular applications, looking at nanomaterials, just a little background about it is when an individual has a myocardial infarction or a heart attack. I've found out that one of the panelists actually did a fellowship in cardiology, so I'm pretty sure he could tell you a lot more about this than I can. This is coming from an engineering perspective. There usually is a blockage in the coronary arteries, and because of that blockage, blood flow and oxygen cannot actually reach that myocardial tissue or that myocardium, that heart tissue, and that tissue will begin to die. When it dies, it's really something that's really interesting about that tissue is it's made out of cardiomyocytes, and those tissues will not be able to reproduce. They will not be able to proliferate like what would happen in the bones or into the muscles. And when that occurs, after a certain amount of time, that muscle begins to atrophy or lose, and then you get ventricular remodeling, which is what you see on the diagram on the right. According to the American Heart Association, over 25% of individuals that have a coronary event will have another coronary artery event within the next 5 years. And the modern techniques that clinicians use to this day, you're looking at angiograms, angioplasties, open heart surgeries, and I found out that last year that there was an amazing robotics lecture going in and looking and trying to fix makes these grafts, really doesn't actually fix the major problem. You have this dead tissue in your heart. And after a certain amount of time, that dead tissue will begin to atrophy. So within this collaboration on the I-95 corridor, we said, well, why can't we try to figure out this thing? So molding tissue engineering and coming up with material science, we decided, well, why can't we use nanomaterials and use the really awesome material characteristics of nanomaterials and see if we can use stem cells and be able to change them into cardiomyocytes and allow them to proliferate and make this cardiovascular patch. And so that's exactly what we tried to do. And so, long story short, we use carbon nanofibers up the range of a diameter of 200 nanometers, and we embedded it in a polymer that we chose that is FDA approved and biocompatible, known as polylactic co-glycolic acid, also known as PLGA. And when it is broken down in the body, it's hydrolyzed, and it's broken down through the water, and it breaks up into glycolic acid and lactic acid. And the body can be able to take this and excrete it out through the normal excretion parts of the body. So again, it becomes biocompatible. What's also really interesting about these nanomaterials is it's conductive. And this is really important when we're looking at the conductive system of the heart and how it beats. It uses the electrical circuits to do this. And what we're able to do is we're able to put these systems together, and because this was a new idea, we weren't sure if this was going to work or not, so we are able to mix these two systems together in a weight ratios, which is what you can see on the kind of the bottom left, on 25% weight ratios. And then we did a whole bunch of characterization. We did a lot of assays looking at growth to make sure the stem cells actually turned into cardiomyocytes. Did the cardiomyocytes beat? Did they begin to proliferate? And again, was the hydrolysis of these materials, was it toxic to the cells themselves? And the answer to all those questions was actually very positive to us. But because of time, I'll show you a really interesting graph. We created a system that actually made the electrical circuitry in the heart, and this was in vitro, so it's outside of the body. We actually designed this whole system, and then we placed stem cells, induced them to cardiomyocytes, and then we were able to electrically stimulate them, and we compared it to non-electrical stimulation. And so on the graph on the far left, the 100% means that's 100% of the polymer that is used, 100% PLGA. On the far right, it's the control, which is just a normal X-class substrate, or as what the previous panelist stated, just a normal cell culture dish to see how the cells grow. What's really interesting is when you look at the middle, which is the 50-50, up to 5 days, non-stimulated, it actually proliferated the cardiomyocyte cells quite a bit compared to 100% polymer and compared to just what you would see in a petri dish. Now, when we electrically stimulated them, they actually proliferated even more. And what's even cooler is when we actually visualized these and watched these, they actually began to beat simultaneously. So we had these beating cardiomyocyte cells that were being electrically conducted through our system, and they're actually beginning to proliferate, which is really interesting. At this time, this research has been moved over to a larger lab at Mass General Hospital, and those smarter people were able to take this over, and at this time, they're actually doing animal studies on figuring out how to take this patch and actually put it over that dead tissue of swine or pigs to see how is it actually going to affect the system. So switching gears a little bit and looking at cancer is one of the ways of reproducing and understanding, how cancer moves is looking through cell migration. A lot of different cells will migrate in different ways and different techniques. But the American Cancer Association, through research, has said if you look at tissue-independent cells, then you can and you watch them how they migrate very similarly to cancer cells. And so there's been a lot of research in doing this. And the cell that we like to investigate at Brown, as well as at Harvard, are neutrophils, or white blood cells. White blood cells are in your circulatory system, and they are actually some really cool cells. So this video on the far left is seeing these neutrophils going through the capillary walls, and they're beginning to roll. And it's rolling because what happens is your body starts a cascade effect when there's a foreign body, and that foreign body then creates a cascade effect, which then releases chemicals. Those chemicals are diffused into the capillary system, to the interstitial tissue, which then activates these neutrophils. When the activation occurs, they roll, they capture, or they attach themselves to the linings. Then they adhere, they spread, as you can see in the far right, and they do something really cool. Looking at these under a microscope, it's really awesome. They actually squeeze themselves in between the cell walls and actually pop out of the capillary wall, and then now they're into interstitial tissue. And then they race through to the gradient of where the chemicals are, and then they engulf the bacteria or they engulf the foreign body. A lot of research has already investigated everything up to the movement of these cells in 3D. And that's the major understanding that a lot of the American Cancer Association really, really wants to know. So because there are no assays that actually can investigate this, we had to devise a whole system. And there are some systems out there, but they're very, very small. We wanted to make this on a larger scale so that we can look under this through fluorescent microscopy and other microscopical techniques. But one of the major problems is how can we measure the movement of these cells? Well, one of my advisors at Brown actually came up with a really cool system called digital volume correlation algorithm. And what it does is that you can take a three-dimensional matrix, you can embed fluorescent microspheres in there, and over time-lapse microscopy, you can image the movement of these beads. And when you measure the movement of these beads through the algorithms and through the mathematics, you can actually ultimately understand the displacements, the strain calculations, the traction forces that these cells are actually moving in 2D as well as 3D. And then you can go to the forces that the cells are doing. So we're able to understand how these cells move through digital volume correlation. And then we actually created this whole system. What's really important about this system is a pure chemotaxis system up to 10 hours. On the market today, there's only a chemokinesis, which means that the buildup of those chemicals in a system begin to build up, and it doesn't create a difference between one side or the other. But this actually has two reservoirs that can continually move throughout the whole system. So when we do that, we're able to embed these neutrophils, human neutrophils from patients at the hospitals. We stain them so we can visually see them, and then we get to have the fun part of actually visually seeing what the cells will do. And so this is a video, a time-lapse video, and the red is the actual neutrophil moving. And that is a 10-minute video. So you can see that cell technically running in three-dimensional space, again, three-dimensional space towards that gradient or towards that chemical. So we said, okay, well, what's actually happening? So we did some microscopy techniques called refractive microscopy, and we're able to visually see the collagen matrix or the matrices and see how the cell is actually embedded into the system. And that's the video that you're seeing. Oop, it went too deep, right? But you can actually, some really cool things. You can actually go in and see how the cell moves in 3D and pop it out. But again, we did this through a whole bunch of other cells and a whole bunch of other time lapses. And what you see with each one of these are these cells are migrating towards the gradient or migrating towards the chemical of interest, which is really important. But then if you look at them, if you look at the one on the bottom right, you could actually see the cell go on the XY axis, and then it moves in the Z axis. Now this was very, very exciting for researchers because most research in cell culturing or in cell-based systems are in 2 dimensions. And there's, if you notice, if you read a lot of the publications, that 2D is very, very different than in vivo or in 3D. So a lot of cell cultures and a lot of scientists are moving into 3D systems. So again, we have this system that is a pure chemotaxis system, as well as we can actually visually see the cell moving in 3 dimensions. So now we have this. We can actually look at the displacements of the cell. We can actually visually see the cell in displacements. And again, this is the cell. It's actually moving in 3D. We can actually see the displacements and the magnitude of the displacements of the cell. So we can actually see how the cell is migrating. And more importantly, we can see the concentration on the cell surface through the calculations we made through the diffusion equations that we created with our system. So this is a cell, and you can see how the concentration is changing. It's migrating towards the concentration, but more importantly, we can see what the concentration is on the cell. So we can actually see what concentration that cell will migrate towards at a different speed. So when we put this all together, we get a really cool image that looks like this. So again, we can see the concentration of this cell in 3D interstitial tissue. We can see the displacements, which are the vectors of the cell, so we know how the cell is moving through the whole system. So we can begin to understand how the cell moves. Now when we use digital volume correlation, we can then get to the strains, and this is the really cool part. This is what knocked a lot of people's socks off when we actually did this. If you notice, so the isosurfaces that are embedded in the cell are the principal strains or the deformation of the cell itself. And what's interesting is a lot of cells, they deform, and you see a lot of the strains in the front of the cell. If you look at a fibroblast migration, it likes to pull. It grabs on something and it pulls. It grabs on something and it pulls. What a neutrophil does, and tissue-independent cells do, is they actually have their deformation in the rear of their cell. So they deform in the rear, change their shape, squeeze, and protrude forward, which is quite counterintuitive to what a lot of researchers were seeing. And so we're showing in 3D that these cells are actually migrating very, very differently than what was proposed or thought of 10 years ago. So just to conclude, because I know my time is short, we were able to think of a different way of trying to help people that have cardiovascular disease or someone that has a heart attack by using a system that was actually very conductive. We're able to take stem cells and to place them and move them towards a cardiomyocyte system. We're able to have it biocompatible according to the system and actually progress it a little bit farther. But some questions to consider is the toxicity. This is the big question according to biologists, is once we have that cell and we put it or we do something after 5 days, 10 days, 20 days, what will occur to the cell afterwards? And then lastly, we're able to create a 3-dimensional migration assay system to investigate how cells migrate in 3D to look at the cancer research. And if you look, we're able to have 6 different variables, dx, dy, dt. We actually can change the collagen density. We can actually look at the concentration gradients and see how the gradients work. And we can have many, up to 5 different outcomes for this. So this is very important because we're able to give this really awesome system. So if you think from an engineering perspective, we created this awesome system where we can say, here you go, biologists. It's now your time to do some even cooler things and to help progress this research. But the ultimate question again is, how is a cell doing this? We're able to show that this cell is migrating very differently than what is expected. But again, the ultimate question is, well, we really don't know how this is still working. We gave the tool, now we need some help from the other side. And I think that's what's really cool about biomedical engineering is it's just not a material scientist or a tissue engineer. It's a collaboration of all these engineers and all these all these different scientists for one common goal of trying to understand, again, how the body functions the way it does. And with that, of course, I couldn't do this research by myself. I did this with a lot of other people and, of course, a lot of the funding agencies. And I would like to recognize that actually quite a bit of this research funding actually came from the California State University predoctoral program that I was actually a part of a few years back, as well as I appreciate the sponsors at the California Distinguished Lecture Series. So thank you very much, and I'll take questions afterwards.
Thank you, David. Next, I think we're going to do a tag team here. I don't know what you guys call it. I'd like to introduce Dr. Fred Burbank and Mike Jones who are both partners in Salt Creek Medical. Mike is actually a graduate here from Cal State Long Beach. So we got our beach up here, and I don't think Patrick, our last speaker, is, but could have been. Okay, thanks you guys.
Okay, so it's time to wake up. Nothing I'm going to talk about is complex. Although I was sort of frightened, I thought special beams came in from the sun through this dome, and I wasn't quite sure where I was coming. I was glad to see it was not a tomb with Egyptians in it. So Mike and I are partners and have been for quite a while. We work out of 3 muffler shops. 1, 2, 3. If you went to a muffler shop, this is exactly what we have. You can throw a small ball over any one building, and we started out with 1, and then we got another one. We got 3 now, and we do primarily blackboard work. No. You can hear me back there? Yeah, I thought so. Okay, so we go from blackboards to scratches on paper to CNC machines, all in these buildings. And for those of you that are, no one's my age here, but those who this used to take a room full of people to make this transition. And you all that are younger grew up thinking, of course, you just draw it in 3D, and then it has a program, and you put it on here, and the CNC machine makes a screw. Well, that was a long process, but we've got that down now. We can make screws and other things here. This thing should start. Not gonna. Can you see it? So here we go. So listen carefully. That's the first piece of information.
Not the last few of these staples, eh?
Okay, so what did you see? Okay. Tricky question. This is a tricky question. You saw some guy hit his thumb with a hammer. A little guy, and he said, ow, and cried to his mother. The other guy swore, but the first thing they did this, okay? So this little lecture series is human stuff, okay? So this is the humans do that. They could have, to 80%, shook the other hand, and it would have helped also, okay? But they generally shake the hand that got hit. But that works too. Okay? So we knew about this because we were both little kids. And I hit my hand. So pain and nothing. You just put your thumb down on the bench and just whack it with a hammer. Okay, it's really hard to hold your thumb there. Okay, it is just, okay. okay? You hit it, and something else happens. You do something else. That pain is less. And this is a human phenomena known as counterstimulation. So you have one set of sensations coming in that hurt or are troublesome. And if at the same time something else comes in, that discomfort is less. Basic phenomenon. So this is a lecture. This is what's so cool about it. The lecture's on natural body function. So it was hard for me to weave this in this lecture because I had no idea what the series was, but that's a natural body function, so we're right on topic. Okay. Now, how many of you folks don't sleep so well at night? Okay, it's common, okay? How many folks have irritable sensations in your legs called restless legs syndrome, which cause you to have a hard time going to sleep or wakes you while you're asleep? Okay, so there's some folks you have, okay? I didn't know I had it until I watched TV one time. Okay, so the drug manufacturers had a drug used for Parkinsonism. They made a buck on that. They thought, we whore this thing out some other place and make some more money. So they stuck it on restless leg syndrome. So I'm watching TV with my wife, and the ad comes on, and this guy's got restless legs, and he takes the drug, and I'm fine now. I think I got that. She says, you're a doctor. You ought to know if you got that. And I said, well, I just thought it was part of getting old. I've been bothering with this for years. So I started thinking about this, and when the drug came out, the experts, I'm not an expert in this, I'm just a doctor guy. The experts said, this is a drug deficiency disease. We just haven't sprinkled enough of this drug in people, and so that's what it is. And it was thought of, you just didn't get enough of this dopamine stuff. So we did not think of this as a drug deficiency disease. And this is the tricky part. There's some concept occurs, some biological concept occurs, and then somebody has to execute the concept in reality, from the blackboard to drawings to parts that fit together and move. So I conceptualized this as a hallucination or a dream. That it started in the brain, and it was projected to the legs when someone's sort of sleepy or asleep. Somewhat like someone who has lost a leg and has a phantom leg syndrome. They have strange sensations about their legs, and they have no leg, but they have a representation of the leg in the brain on a specific place. There's a little homunculus in there that shows hands and tongues of exactly where the body parts are represented in the brain. So we thought this was a phenomena which was more like hallucinations or a phantom limb or a dream, and less a drug deficiency disease. So projected to the legs. Instead of being in bed and having almost no sensations coming from your feet and your ankles and all that kind of stuff, you stand up, and now you have real stimulation, real sensations coming in. You have tension on your ankles, your muscles are doing something in your leg. You've got some skin phenomena on your jammy. You've got real things, and like a dream going away, the phenomena goes away. You go, I'm okay. I'm going to lie down, and then I'm going to lie down now again, and it's going to go away, and oh no, there it goes. But I'm going to do this with my leg, and then you have to stand up again. So it's a cycle that deprives folks of sleep. But if we could do something to cause leg inputs, because that's where the projected phenomena seem to be going. Some people have in the arm too. You could keep someone in bed with these real stimulation from a man-made thing by engineers, then maybe you could lie in bed, have this thing do something, provide sensations to you that were pleasant, and then kind of went off slowly, so you could focus on those, not focus on what you're feeling, and go back to sleep. So that was the task, which was then given on a blackboard to Mike. And we've done many projects, and this is just another, okay, so here we go. So Mike's going to do the rest of the task. So here is the...
All right, so Fred realizes he's got restless leg syndrome. My wife has restless leg syndrome, and I'm actually surprised that we had only a few hands up here, because it's about 5 or 10% of the population that has restless leg syndrome. We started the project, we created a sleep lab in one office of our approximately 6,000 square feet buildings. We recruited through newspapers to bring patients in who had restless leg syndrome and to try out a bunch of different ideas. So on the blackboard, Fred and I sat down and thought, what are all the different ways that we can provide stimulation to a patient while they're in bed? I said, well, we could put a heating pad under their legs, we could put an ice pack under their legs, we can provide vibration through a massage pad, we can put pneumatic compression sleeves on their legs and sequentially squeeze them. We can put TENS on them. We can give them a light to look at. We can give them a sound to listen to. We can put a chemical, icy hot. We could put a thermoelectric cooler under there. But there are a number of different things we went through and said, all these would provide some level of counterstimulation. And so for the next portion was, and that was for our patent, all the different ways we can come up to do this. The next one was, well, we really need to sort this out so the patient can stay in bed. Don't have to get up and walk around. They can literally just, whatever they're doing, put their legs on top and try to go back to sleep. Needs to be really easy to apply so that they can stay in bed and go back to sleep, not get up and walk around. So we threw out all the really messy ones. Chemical application, TENS, heat and cold, and we designed a test matrix to evaluate these devices up here on these patients. So we had, I'll start in the upper left, high frequency vibration. So this is a massage pad on steroids. It's a big counterweight on a series of 6 electrical motors. Turn it on. There's a little knob up there that allows the patient to vary the frequency from about 30 hertz at the low end to 60 hertz at the top end. Second one was a low frequency vibration. So we had a linear coil. Had it set up to run from about a half a hertz to 10 hertz, but a much higher amplitude for the patient to look at. We had another pad that looks the same. In one sense, it had a little LED light in the middle of the pad. So the patient could put their legs on it, look at the light, turn the knob, light got brighter, light got narrow, got reduced. They could actually concentrate on that, see what they like. Same thing with sound. We had a little sound generator. They can make it go a little higher frequency, a little lower frequency. We had one that we thought was laughable, was a fixed light. Plug it in, turn it on, its light comes on, the patient doesn't have any control of it at all. And then we tested pneumatic sleeves, which the patient put on like a pair of socks, and then a vibration cuff, sort of the same thing, but the vibration was delivered right to the site of the patient's maximum symptoms. So we introduce these to the patients in random. We interview the patients after all of them. They end up ranking them. And what we found after all of this work was high frequency vibration was the most readily accepted of the various forms of counterstimulation. The low frequency was less accepted. It was pretty dramatic. And basically, the variable sound and the variable light were almost identical to low frequency vibration. And then pneumatic sleeve, fixed light, and vibration cuff were essentially, they were another order of magnitude down in terms of how patients thought they would, how the patients were able to use them as a counterstimulus.
Okay. So we then ran 2 studies, two small studies, and we integrated those and compared those to a substantial number of drug studies through a technique called meta-analysis, which is one of the kind of the modern major building blocks of clinical statistical analysis of all sorts. So if you can then compare indirectly things which would be very hard to compare in a single trial. So we compared our device to active shams, and we used for one trial a noise generating sham. Another device was our device versus a light, a cold light, an LED, which you could make it brightest. So ours was against a sham, and the drug studies were against pills that were inactive nothing pills. Probably the wrong placebo, because all these drugs make people really sleepy. And so an hour after you take one of these drugs, you can't read the paper. So it's easier to break them. And we later on compared our shams with theirs, and they're about the same. So we were better than our shams, and we are about the same within the measurement errors as the drugs currently approved by the FDA. So we have a device which can be used in bed. You can put it, turn it on, runs for a while, turns itself down, you go back to sleep. And we've shown that it is equivalent to the current gold standard for drugs approved by the FDA and better than a sham. Now some ugly stuff. I hate to do this to the young children here. All those under 21 will have to leave, but the ugly stuff is reality in the medical device world. Have any of you heard in the news that the Obamacare rollout's not going perfectly? I read something about that, though I was told it was going to be perfect, and I would love it. So here's the ugly story about medical devices. As of today, we have spent 5 years and 16 days in the FDA. Okay? We applied for this 5 years and 16 days ago, right now. We spent 4 years and 9 months to get to a rejection, went back to the FDA, met with a big room full of folks, and said why they were wrong. And they wrote back a month later saying, you know what, we're wrong. But let us kind of look at your data, and if we can add up the same way you do, we'll approve you. So at 5 years and 16 days, you can't start a new company that has a 5-year, 16-day gestational period and lives. Medical devices take about a year to get going to where they can do something. And during that year, you get through the patent office and through the FDA. That was the old world. The new world is the government's broken. We have almost exactly the same timeline on the patent office. So medical devices, at one point, were kind of simple. This is a wiggly pad. This is really simple. Okay, so this is not something. A similar sort of thing can be purchased in a drugstore. If the government is inefficient like this, you can't fund small companies because they can't go 5 years where they start to have some sort of income. They will die at year 1. So all the nice stuff these young guys are going to hope to do, it's all predicated. It's predicated on being able to get through that. We were assigned to this branch, the restorative branch. Then we got assigned to the ophthalmologic branch, the stimulators. And we went back to the restorative branch, and we went to somebody else. We spent 5 years to have them say, you know what, we didn't do this right. Your analysis was perfect. So all the science stuff is interesting, but it interacts substantially with the financial world. And these guys will learn about that as they raise money for their little companies. And then it falls into the great black hole now of the federal government. And it's a major concern that if the government doesn't do its stuff in a reasonable period of time, no matter what they do, they will fail. They can't fund this. I'd rather fund a storage depot, U-Haul storage, than I would one of my own companies now, because I can't get through the government. Okay, thanks guys for listening.
I'll tell you, I didn't really realize this, but this is a great lead-in. We have Patrick Ikahara here, and Patrick is with Applied Medical, but he is going to be talking about, how can we get stuff done? Patents. Maybe you know how to get through the government. Maybe you can give them a hint. I tell you, it's going to be interesting.
Yes,
So it's all yours. Good luck.
Thank you. I mean, I didn't think my topic is going to be all that relevant, but maybe now it is, based on that kind of lead-in. It's kind of a harder act to follow, and I'll try my best to kind of maybe respond to some of that. But anyway, so I'm going to talk to you kind of, again, kind of off topic, but I guess is now more relevant. Now it's about patenting. I'm talking about one of the major forms of intellectual property that we use I'm a patent counsel for Applied Medical, and we use it to protect our market shares, to get funding, to get, to protect our, what products we build and try to maintain our R&D dollars. so patenting body functions, like how the heart works, how it pumps blood, that's something we don't do. And that's also something you can't do. It's prohibited. So what is patentable is you can patent devices that replicate those functions, like artificial hearts, for instance, prostheses or other biotechnologies. We're more focused on patenting devices that assist in the repair of the body, so more like surgical instruments. So I'm going to talk kind of more in general about the challenges that we have with patenting. And maybe I'll address some of the issues here. But there's a lot of information out there. I mean, there's a lot of information. A lot of some of it's misleading. Some of it's hype. Some of it's, trying to find that right information from a patent's perspective is really, really difficult. It also ranges through multiple issues. I mean, there's not just the technology issue. I mean, there's a lot of that still has to be considered and understood because it is very technical. There is it can range multiple disciplines. You heard here that there's mechanical issues, electrical issues, biology, clinical issues. There's a lot of that, but there's also the business perspective of it, the business reality, the financial reality of it that you have to consider and understand to do this. Because patents have a long lifespan, and they take a long process. I mean, you've heard it here. It could take 5 to 7 years. So you have to be able to patent and effectively patent what you can with that in mind, with that long lead time in mind. So you have to be able to find that kind of information, gather that information, understand it, and have kind of that future sight or that foresight to be able to capture that and to basically understand it. And hopefully in that 5 or 6 year when you do get your patent, that you're right there. You have the ability to cover your product, to protect your markets. We, as Applied Medical, we've been fortunate that we were able to get past those first year's marks. And patents, for us, gave us the opportunity to become from a 30-person company to a 3,000-person company. I mean, we have hundreds of patents now. So patents for us is we're pretty fond of patents. We kind of like patents. So, okay. Anyway, I don't know what that is, but patents are basically a grant from the government. So it's only as strong as the government is. So it's only as strong as the government's willing to back it. So the patents, if the patent system fails or the patent system is having problems, right, the patents aren't going to be as strong as they are. But it is still a government process, so it's there's a lot of red tape. There's a lot of administrative formalities you have to go through. You have a lot of hoops you have to jump through. And it is just like the FDA. There's a lot of issues you got to go through and go through it. Kind of the unfortunate part, because a lot of those administrative and formality issues you have to address, if you make a misstep, you make an error, even if it's innocent, it could take you months to kind of correct that, years to correct it. And at the end of the day, it could cost you patents, it could cost you rights based on that little administrative misstep. And so it is still kind of a very daunting prospect of trying to go through this. I mean, one high-level patent official kind of explained the patent office to me. It's like the U.S. Patent Office. It's like one giant office, right? There's only one in the United States, but there are thousands of patent offices within that office. So you have to navigate all that and try to get a patent at the end of the day. So the other challenge is that there's a mountain of patent out there. There's 8 million patents issued today. There's 500,000 filed every year. There's 500,000 pending in the office right now. And so there's a lot, and that's just the U.S. So this is a global system. So in each country they have their own patent system as well. So there's a mountain of patents out there. So you have to consider all that and understand all that. And patents, unfortunately, are kind of an archaic document. I mean, we have to take one of these presentations we got here and condense it into a document, make it understandable to anybody, because it could be anybody that's going to read this, and then try to cover it and protect it for a future of a 5 to 10 year kind of mark at it. And so this, and then top that all off, you have to have a bunch of legalese and a bunch of layers on it to make sure that you're anticipating the changes in the patent office or the laws as it changed over the year. And this is another thing about the patent system too. And with patents in general is that it's constantly changing. The knowledge base is constantly changing. Not only from a technical standpoint, but from the patents or the patent law standpoint. I mean, at one point it was really static, and there wasn't much change in the law, but now it's in vogue to change patent law. I mean, there's patent reform that's being called out there, and it's an easy issue from a political standpoint, because there's something broken and everybody wants to fix it. So there's patent reform on the way, and I don't necessarily that's a bad thing. I'm just saying that you have to still anticipate all that. You have to anticipate all that change if you're going to have to work in the system. And it's not just isolated to the U.S. It's also in Europe. Europe is probably it's the closest it's ever been to having a unified patent system. China and other countries like that are getting more and more robust system to try to make patents better in their countries. So the other challenge is time and money. I mean, it costs a lot. I mean, it costs $10,000 to $20,000 just to file a patent or get a patent in the United States. If you try to go internationally, it's 10 times that. If you want to go through multiple countries, it's a pretty expensive proposition. And trying to investigate it and clearing through patents is also significantly high in the money category and also the time category. Also, patent lawsuits, trying to defend it and enforce it, it could cost you millions of dollars. Trying to defend it. Some of the highest patent awards have been in this medical device industry. Just recently, Boston Scientific had to pay $2 billion to settle a patent lawsuit. And so again, the stakes are really high, and it costs a lot of money. I mean, not money, just time. Time, mentioned here, this could 5-7 years of your life that you get. You have to invest in trying to get a patent. So there's the other challenge was there's a tendency to kind of ignore it, try to hope it goes away. You kind of bury your head in the sand and just say, we'll deal with it some other time. We'll wait for the last possible minute. And kind of the problem with that, of course, is that if you wait till the last minute, then your product may not be as good. You may not be able to respond as well to a patent infringement letter. You won't have other options to take on it because you hadn't really considered it all. The other problem is in this industry, you really can't avoid patents. I mean, a lot of these folks will run into patents. They'll run into patents that they have to go through and then figure out how to get around it or get through it. And so patents are unfortunately, really prevalent in this industry. And also they're highly enforced. I mean, there's established companies that will enforce their patent rights to keep their market shares, to protect their R&D. So it's a highly litigious industry or area too. So it's something that is something you really just can't ignore from that standpoint as well. The other thing too is that once you make your product, once you're out there and you're doing well, then you get knockoffs coming off. And the only way you may be able to protect it is through your patents. So I complained a lot, and it's kind of my last, this slide I'm not really going to talk a lot about, but there's kind of some considerations kind of maybe to address what, the challenge is kind of with the patent office. I think probably the thing maybe to take away from this is that if you can treat it objectively, treat it like just any other business tool, I think you're better off kind of overcoming the challenges of the patents. Unfortunately, in this medical device industry, it is a highly litigious area, and it's also something that you really will have to run into at one point or another.
Thank you, Patrick. I don't think you fully solved Fred's problem yet, but maybe you can give him a little advice at the end here, where to go. Okay, we're going to have a question and answer session. People want to come up, but first of all, I just want to mention that our next lecture is going to be April 24th. It's Thursday, so remember that date. That's going to be another, I think, fascinating topic. So. April. Oh, I didn't even look at that. I know the date. April 24th. Oh, it says 24th is close. Thank you, Dean. Okay, what I'd like to do is open it up for questions. People have questions? Yeah, Mike, go ahead, start it out.
I've got a question for Adam and also for David. Adam, in your tissue engineering, as you're looking at taking this product out, I'd like to know about how long do you think it's going to take so it gets to be widespread, and what kind of applications do you actually see in the next few years? And then for David, on the transport mechanism you're talking about, as you figure the transport mechanism out, how is that going to impact either the treatment or detection of the diseases, the cancers that you're really looking at?
That's a great question. So for us, speaking to patents, we filed our patent in December of 2009, and we still don't have it approved yet. So yeah, it definitely takes a while. Fortunately for us, we have an easier path to market in that we can go through the cosmetics route. So that gets us a little fewer hurdles in terms of what we need to jump through to get to a product actually being on the market. So we're expecting to start human trials in about the next year to year and a half. And then from there, hopefully being able to progress a little bit quicker within, I would say, maybe 3 years of trying to get onto the market. But again, as these guys can attest, you can never tell how long that FDA review process is going to be. Fortunately, there are other products similar enough to ours that we can try to go through the device route. But if we have to go through a PMA or a full-blown FDA review, then it'll definitely take a lot longer.
That's a really interesting question that you stated, because I wasn't able to talk about some of the other aspects. But a lot of the cancer research that's been being investigated is looking at the cells and looking at integrins. And integrins is the way that cells can talk and connect to each other and then move to certain applications. And research has shown in a lot of tissue-dependent cells that there's a way that you can block these integrins. And if you block these integrins, the cells won't move or migrate to the desired location that they want to go to. But what's really interesting is in a 3D world, if you do this integrin blocking or blocking the way that the cells can attach and talk to one another and move and migrate, in tissue-independent cells, they've shown that these cells can actually continually move to the desired location. And so past research has been investigating with billions of dollars looking at integrin blocking, or just blocking the way that the cells can communicate and talk to one another. And what we've shown is that maybe that's not the right way of looking at this research. Maybe we should look at a different aspect and say, all right, maybe there's a different mechanism that the cell is using so that it can actually migrate or for example, the cancer cell can metastasize to the new location. So the question is kind of really opened, is we are kind of proving that what's not really proven, but we're showing that what a lot of research has been doing is kind of, I don't want to say incorrect, but maybe it's not the full scope of the measure of the issue. We're saying, well, this is the issue, and maybe we should look at a different aspect. And maybe it's kind of a hybrid of integrin blocking or blocking the way the cell can talk, as well as maybe blocking the way that the cell moves interstitially or moves within the cell itself. So it's a very good question, and it's still open, and that's why we need help from biologists to say, all right, this is kind of the wrong direction. Maybe we're going, maybe we should look at a different direction.
Right. Thank you. Other questions? Yeah.
It's a question for Adam. It seems as though you've got some cellular differentiation there, which is very, very specific to your ECM, which means that you've got some nuclear signaling going on and then fatty acid synthesis. Do you know the mechanism by which you get signal transduction?
Yeah, that's a really good question, and something, again, that the biologists help us out with. So I didn't show this data, but we have looked at how adipose-derived stem cells, so the resident stem cells present in fat tissue, interact with our matrix in the laboratory, and so in vitro. And we've looked through some of the mechanistic pathways of what's driving those cells towards some of this, the differentiation in terms of producing lipids and taking on more of an adipocyte lineage. And A lot of it has to do with how the proteins they're seeing. So we're providing them with ECM components that are characteristic to adipose tissue. And part of it is also regulated by stiffness as well. The cells can sense that they're on or within a material that's about the stiffness of adipose tissue, and that can drive them more towards that type of a lineage too, versus if they're on something really hard, they go more towards a bone lineage. So we speculate that that's a little bit of the mechanism that's going on there, but we have more work to do to determine that for sure.
I think that's just a fascinating topic, and it's just the research that's going on. If you can really figure out how the cells move around in there, and I mean that's really going to go a long ways to helping cancer patients know what might be coming next, or be able to even stop it from moving around. So I think it's just great. Other questions? Yes, sir.
This is directed towards Patrick and the challenges that Adam is going to be facing that Fred pointed out. One of the things that I've noticed is it's really rough being an entrepreneur. I've been to a number of auctions picking up equipment from failed companies, but part of that is one of the assets of a failed company is the patents, and some of these are falling into NPEs, non-practicing entities. And I was wondering how Patrick has been dealing with those.
Maybe kind of also respond. From a patent perspective, like from a medical device perspective at least, we don't run into a lot of NPE problems. We don't, typically it's because the industry is relatively, at least the industry for the medical device industry, it's historically has a lot of knowledge base behind it. That's not just pure patents. So there's a lot of force that you kind of bring behind that you don't run into a lot of the NPs. I mean, and for those who don't, they're called non-practicing entities, or also known as patent trolls to some people. So they're basically licensing companies. They're basically companies that just acquire patents and then try to enforce it on the non-suspecting person or non-suspecting business. It is really relevant in a lot of consumer electronics devices. I mean, Apple probably gets thousands of these every day. So it's really prevalent in that kind of industry. It's not so much in our industry, but that could also change as more and more of these failed companies come up. But right now, we don't see a lot of impact of it. But there's a lot of legislation going on. There's a lot of things going on that may curtail a lot of that stuff. But it also kind of goes to one of the things with patents is that if you look at Kodak, for instance, Kodak had a bankruptcy, and they sold their patent portfolio off for $500 million. I mean, for 1,000 patents. So these things are pretty valuable, and so they're going to constantly be snapped up. I mean, they also had other assets of like plants and electrical power plants that went for like a fraction, like $8 million. And it's really a fraction. In other words, it wouldn't surprise me in the future that we might run into these NPE problems. The thing is, though, I think, like from our medical device standpoint, there's a lot of historical stuff that we have to back up kind of what we're at, and there's also a lot of testing that goes behind the scenes that kind of help this define what we would call a priority to kind of combat some of these more frivolous kind of things. But it's still something that we don't have right now have a problem, but it could eventually become a problem.
Okay, thank you. Other questions? Okay, I got one for Fred, because I was listening to you, and at the end, when you talk about over 5 years, are you able to, the difference in having to take pill and, or some type of medicine to get through this versus a mechanical device or something you attach or whatever, I think is a savior to a person, not having to take those drugs. And are you able to get out there and market your device at all, or not until you get the approval?
I think we can go to Australia soon. We've got patent approval there, and I think we can get through their equivalent of the FDA. We can't do anything in the United States as an economic entity until you have a patent approved and you're through the FDA, because you can't get financing to be a company. There's the minimum requirement for financing is to have both of those done and have them done well. Without that, you're just in your garage tinkering. So you have to go through both those agencies from the government before you can get out and then actually start to really do real work. That's an absolute bedrock requirement. Because mainly for small companies, you want to be bought. Okay? Small companies don't become big companies. Big companies eat small companies. And they don't eat small companies that don't have patents and have the FDA approval, because they don't do well prior to that. So all big companies in the device area grow primarily by eating small companies. That's how they grow.
Well, what can I say to that? Okay, we have another question. Good.
Question for David. So the U.S. Navy has put a lot of money into the hunter-killer submarine program, where you're trying to detect a signal which is static in three-dimensional space based upon a diffusion gradient. And the models that they've been using are similar to the ones that moths find their mate along a very, very diffuse diffusion gradient. And all of these types of organisms use a spiral track, and then they pick up the angle, and then they follow the diffusion gradient. And that represents the most economic pattern to find a point source. So do your cells spiral?
For some reason, I thought someone was going to ask that question, and because it's actually been asked before. And the short answer to your question is yes. These cells, they don't directly go straight to the gradient system itself. They will actually, I don't want to say feel, but they move around in a spiral technique in the XYZ axis, and then they find that perfect angle attack, and then they technically actually speed up to that position of the gradient, which is really interesting because it's, I don't want to say Mother Nature has designed it that way, but we are looking at how moths do it. There's another individual that's actually in my lab that is funded by the Navy, and he does a lot of sonar research, and he's making biosensor sonars, and he's looking at how bats listen to sonars and how they get to the direct angle attack. And they've actually seen that, again, it's this spiral technique where it's a sonar, somewhat of a sonar gradient in three-dimensional space, and then they actually will attack the prey or attack or go to the food in this particular direction. Now, for those researchers out there, the question is, why is it a spiral gradient? What is it so significant or so special about that spiral gradient that a lot of different biological systems want to go to that direction? And that's, I think, a bigger question that needs to be answered.
There's been some research which shows that mathematically that's the most ergonomic tracking device that you can use if you're trying to pinpoint something three-dimensionally. So it's a matter of ergonomics. And that's why over 3.4 billion years of evolution, all biological systems use it.
Yeah, I mean, we can wait another 3.4 billion. It's better now. But no, I totally agree with you. It's very interesting just to see it in different biological systems, even down to the cellular level. You'll see this spiral movement.
Okay, I don't know if there anybody else. I got one more question too, and this is something that I in my experience, basically all the people really working on patents are lawyers or have at least a law background to help them through that. In fact, it's interesting. I actually had a gentleman work for me one time in engineering who also had a law degree, and he had been doing patent. He was a patent attorney for about 5 years doing engineering patents, and he decided he wanted to come. I don't know, maybe the headaches or whatever, but he loved coming back to engineering, and he's a great engineer, so I was glad to have him. But it's interesting. Is that really, you almost need a law degree to really do, or what type of a degree, and what helps you really become a patent attorney? What do you need, the background?
This is just too much fun for me.
So we travel from California to Washington, D.C. in June, Mike and I. And we get greeted by someone in that big building that was shown there. And they didn't realize there was actually two of us coming, so they had to find an extra chair for us. And then the woman we're talking to, I think she finished college the year before. So she's a college graduate who goes to patent school inside on-job training. And that's her background. So it would be a treat to have the patent office have lawyers in it, but they don't. They have folks who've gone on-job training to then learn patentese, and that's who the examiner is. Scary.
The reason why I'm not really arguing is because I do agree with them. I mean, I agree that there's a lot of frustration, and because I'm in-house, so I actually work for my company, Applied Medical. So it means a lot more to me that I don't get a patent, right? So I do take it a lot more personally, even though I shouldn't. But yeah, I mean, it is frustrating running into less than experienced people trying to explain it to them. And as a lawyer too, there's rules and laws that you're supposed to follow, and then some of them just don't follow the rules. Just don't follow the law. So you kind of, but you also, there's laws that prevent us from lashing out at them. So, so we have to, be simple about a lot of these things, and we try also not to. There's a very complex issue. I mean, patents themselves is a really, really complex issue. I went through a bunch of challenges, but it's a really complex issue with a lot of issues out there. It's really complex. There's little nuances here and there, and working in it is even harder and has even more problems. But I mean, you take the U.S. Patent Office and you compare that to the European Patent Office. They're night and day. You have PhDs people, you have really, really smart and intelligent people on the European patent level. That's really, really different. And I think part of that is largely because of the way they're treated. I mean, they're not treated based on quotas. They're not basically treated on, how many patents do you allow today? What's your backlog in it? I mean, they're actually treated as this is a great career opportunity for you to learn all about patents, learn about, so PhD guys are coming out that President of the Patent Office came out.
You have to remember that Albert Einstein's first job was in the patent office, and it was such slow work he could figure out relativity. So this is I'm pretty sure the lady I was talking to is not going to do relativity research during her lifetime. So having the brighter person across from you is a real benefit for an applicant. It's true for almost all endeavors. That your opponent being very good, you'll come to a better end than having a disparity on either side.
Yeah, that's great. Any other questions? Okay, Dean's coming back up here, Dean Golshani, but I just want to thank our speakers, and it's just great. Dean, all yours.
Thank you. This was a great panel. It was informative, enjoyable, and engaging. Thank you. I learned a lot. I can be in innovation business, come up with lots of great ideas, go out for a patent, wait for a while, fight for a while, and then you get the patent, then go for FDA approval. And eventually that comes as well, right? I wonder, well, let's assume we have these two. What's the next step? I think that's the most difficult one. We have to have a panel on that one, how to deal with the insurance company paying for it. How come we didn't cover that one? Anyway, thank you. There are little gifts that are prepared for the speakers. I'm going to ask them to come on down and receive these little gifts. Thank you very much. Thank you, everyone. Make a note of the date, April 24, and look forward to seeing you again.